Okay, quick overview of the components of milk. Well, we know there's fat, we know there's protein, we know there's sugar, right? It's got everything that a young organism can need. Let's break it down though. Okay, first look at the figure. This is from your text. We see the mammary gland over on the upper left, and we zoom in and finally in on the cellular structure itself, and we see that, okay, this is a normal cell. It's got a nucleus, it's got an endoplasmic reticulum, where proteins are made. It's got little mitochondria where the energy is produced, but it also is full of oil droplets. These are insoluble blobs of triglyceride, but they bud forth from the bud forth from the cell, and as they come out of the cell, they're covered with the components of the cell membrane, glycerophospholipids, as I said before. That's why I'm referring you back to low-density lipoproteins. Those cellular membrane components contain emulsifiers, and those emulsifiers are important because they help keep what would otherwise be an insoluble particle soluble in the milk, which is a water phase, an aqueous phase. So there's lots of fat, and it's emulsified. As much as 5% fat, 3 to 5% fat in cow milk. There's also protein. Well, let's break it into two versions of protein. We're familiar with curds and with whey. The curd proteins are the ones that they aggregate and precipitate out of solution when we make cheese. They're also referred to as the caseins. We'll look closely at them in a few minutes. They're there to provide a nutrient for the growing organism. The caseins assemble into large-scale micellar structures similar to what we've seen for the lipids. I remind you that the micelle is a structure that hides hydrophobic or oily surface area on the interior and exposes polar surface area to the exterior. The caseins also play an important role in solubilizing what's called colloidal calcium phosphate, which otherwise would not be terribly soluble. So in milk we see a much higher calcium concentration than we should, and that's because those casein micelles solubilize and dissolve those and bind to those calcium phosphate molecules. Also, we'll come back to that, but then let's turn to whey proteins. Beta-lactoglobulin is one component of the whey. There are many others that we'll look at in a few minutes. The function of these is not always quite as obvious as some of the proteins that we talked about in the egg white. They are nutrients, but in terms of cooking, they help promote foaming. Right? When we denature these proteins, this helps stabilize the foams that we form there from them. So fat, protein. Of course, there's plenty of sugar in milk as well. Cow's milk is as much as 5% lactose. We talked about carbohydrates in the first week, and I remind you that carbohydrates come in a variety of flavors: monosaccharide, disaccharide, trisaccharide, polymers of carbohydrates. We talked about those. This happens to be a structure of lactose. You can see that it's a disaccharide composed of 1 unit of glucose and 1 unit of galactose. They're linked, for the biochemists in the audience, through a beta 1 to 4 glycolytic bond. Not so important for us right now. The differences between these two molecules is subtle. And all I'm going to do is get you to look closely at the structures and look at what kind of bonds we have drawn here. The solid triangles that are coming out from those vertices, imagine those bonds as coming up out of the plane of the screen from you, and the hashed bonds are headed back into the plane. So I want you to compare the two molecules and see if you can identify where the differences are. Subtle differences, but they are important. In any case, we have to break it apart in order to utilize it as an energy source. We'll come back to that. Sweetness index, 0.3. So lactose is less sweet than sugar. Now, before we talk in detail about milk and its chemical properties, let's look at some definitions. Think about what emulsions are and what emulsifiers are. Immiscible liquids are combined, so that's what an emulsion is. Emulsions and colloids, I'll mention the word colloid, are similar. Emulsions, again, focus on two liquids. In a colloid, we're referring to a solid dispersed in a liquid phase. We have two different ways to think about emulsions. One, the dispersed phase, that's the phase that's whipped into tiny droplets. and dispersed throughout the continuous phase that bathes and flows around all of those tiny droplets. The volume of the dispersed phase, that is the amount of those droplets relative to the continuous phase, by necessity increases the viscosity or retards the flow of that continuous phase. So that makes the solution or the emulsion a little bit more viscous. And that can be useful, as we'll see when we go to discuss creation of sauces. How does the dispersed phase stay in solution? Well, there's a force exerted against dispersing that phase in solution, but emulsifiers act to reduce that force and allow that dispersed phase to stay suspended for long periods of time sometimes. So we know what emulsifiers do. They interact with both phases. Recall the polar head group and non-polar tail group, allowing the emulsifier to interact both with the aqueous continuous phase, perhaps, and with the oily dispersed phase. So this relieves the surface tension and allows those droplets to remain in solution. I just wanted to show a few examples. 2 oil and water and 1 water and oil emulsions. Very interesting. So we didn't talk about this last week, but mayonnaise makes use of emulsifiers found in egg yolks. We talked extensively about egg yolk emulsifiers. So first, though, we start with oil droplets. That's our dispersed phase, and we're whipping those oil droplets up into tiny, tiny droplets, increasing their surface area dramatically, and again, retarding that flow of the continuous phase, which is water, and perhaps the proteins that are present in the egg yolk. Lecithin, that egg yolk emulsifier, allows us to maintain that emulsion over long periods of time. You're aware that you can leave mayonnaise sitting in your fridge almost indefinitely and you won't see any separation. And obviously mayonnaise is a very viscous emulsion. Another example of an oil and water emulsion is milk, of course. The fats and the caseins, those provide us our dispersed phase. Water and whey, that's our continuous phase. And as I mentioned before, the emulsifiers are phospholipids that are associated with the surface of the fat droplets, and the whey proteins themselves can act as emulsifiers, as we'll see. Milk also provides an interesting example of a water in oil emulsion. In this case, the whey becomes the dispersed phase, and the fat globules that are broken by mechanical stress, as they're whipped up, the fat globules are broken apart, though they form a continuous phase. So in this case, the fat has to be very concentrated so that that can serve as the continuous phase, and the droplets of water are what get dispersed within it. We'll talk in detail about that. But milk is an interesting substance because we can generate both kinds of emulsion, although oil and water are by far more familiar to us in the kitchen. Let's talk for a moment about lactose intolerance. This map is from Wikipedia. It's a pretty map. It's not terribly well founded in the scientific literature, but it does serve to show a point. We are all familiar with lactose intolerance, or many of us may be familiar with lactose intolerance, and that is just the discomfort that is generated when we consume a high quantity of lactose, gastric distress, if you will. This map shows in red the populations or the regions of the globe where individuals suffer greatly from lactose intolerance, and then in green it shows us where there is no lactose or limited lactose intolerance. Now, again, I won't bank on the precise numbers shown by this map, but it's illustrative. First, lactose intolerance. Well, it's a kind of a funny phrase. If we think about what developmental stage an organism needs to consume milk, we'll realize that it's really infants that need to consume milk. They need as their principal nutrient source their mother's milk. After about a year or two for human infants, there's no longer a need for exclusively milk as the nutrient source. Instead, the organism is eating lots of other kinds of food. So it makes sense then, from a biological perspective and an economics perspective, that we might limit the production of the molecule that's involved in digesting lactose. That enzyme is lactase. It's an enzyme that breaks lactose up from that disaccharide into two monosaccharides that can be consumed. Usually, after weaning, that gene, the gene that encodes lactase, is downregulated, and as the growing organism switches to other food sources. So lactase is also known. Here's just some again for the interested biochemists in the audience, also known as lactose fluorescein hydrolase, or LPH. It is the class of enzyme is called a beta galactosidase. Remember I said that the galactose and the glucose were linked by a beta 1 to 4 glycosidic bond. It's encoded by the gene LCT. You'll note that here's some more nomenclature stuff for you for those that are interested. When we refer to a gene, we usually italicize that name LCT, and when we're referring to the protein that's produced by the gene, we don't italicize it, LPH. It's a little confusing, that nomenclature, but the bottom line is that lactase is, in most cases, only active, only produced in the organism during the period when the organism is usually exposed to high concentrations of lactose, that is to say, as an infant. So it might be more fair to call it, instead of referring to lactose intolerance, the inability to digest lactose, we might think of lactase persistence as being more unusual. That is to say, those individuals that continue to produce lactase after their infant period, that's more unusual. So what we see in Northern Europeans is a mutation that eliminates this downregulation, this infant or post-weaning downregulation of lactase, and that was enriched in the population. In some populations, and especially those populations who came to rely heavily on milk as a principal food source, and I'll elaborate on that on the next slide. This is a complicated slide, so let me go through it slowly. Lactose intolerance, the idea is gene culture co-evolution, so the genes and the culture co-evolve. Heavy reliance on dairy in the Neolithic North Central European diet could have led to variability in herd genetics, especially for the genes for milk proteins. So it might seem intuitive to you to imagine that as those populations in North Central Europe came to rely more and more heavily on dairy as a food source, that their genome would respond to that effectively, to that change in environment. That is to say, the population selects for individuals that are more able to cope with that new food supply. So that's a perfectly reasonable statement, but let's also focus now on what happens to the cows, because that's important for the maps that I'm about to show you as well. So you could imagine that when you're trying to develop cattle herds as a milk supply source, that might lead to genetic variability. You're seeing a whole slew of new traits that crop up from which you're going to select the ones that produce a high quantity of milk. That's the hypothesis presented in the papers that I show you down here on the bottom right, and this is figures from one of those papers. Let's look at their maps and then we'll come back again to the point. On the left we see distribution of genetic diversity among 70 cattle breeds. So these scientists went out and genetically screened many cattle all across Europe and looked for the highest degree of genetic diversity in that population. And then they looked at the distribution of lactase persistence in the European population. So remember, what they're looking for here is that mutation that keeps lactase turned on throughout the life, or at least for longer in the lifetime. And what you see is that they're strongly correlated. That development of genetic diversity, selection, if you will, for high milk volume production, correlates very well with that lactase persistence. So the conclusion that they made is the mutation that leads to lactase persistence is much newer than the normal non-persistence allele. So I'm putting normal in quotes because anytime you start to discuss what's genetically normal and what's genetically abnormal, that's a very sticky topic. But fair to say that the non-persistence is more common across the human population, and the lactase persistence is less common, although quite common in Northern Europe and now in the United States. Just again, here's another detail for those that are interested. The best candidate mutation, there's several mutations in lactase regulation and the lactase gene itself. C to T 13910, that is to say it's 14,000 nucleotide bases upstream from the LPH start codon, the region known to bind the transcription factor AP2. That's a lot of jargon there. Let me break it down. It looks to be that the mutation that leads to lactase permanence is upstream before the protein coding region of the gene in a regulatory region, 14,000 bases outside the protein region, the protein coding region itself. Okay, so a lot going on there, but then only to show you that there is some science going into trying to understand when and how lactase persistence developed in the Northern European community. And again, the idea is that the lactase persistence gene, the C to T minus 13910, developed relatively recently, maybe 5,000 years ago, whereas the non-persistence allele is much more widely distributed and much older in the human population. So the genes evolve with the culture's choice of food, production. I wanted to dedicate one slide to the to milk nutrition and the vitamin components that are found in modern milk, focusing in on two fat-soluble vitamins as opposed to the most of the B vitamins, which are water-soluble vitamins, vitamin A and vitamin D3. Vitamin A is known as retinol. You see its molecular structure here in the upper right. You might notice, if you're paying attention closely, that it looks like half of a beta-carotene, or similar to one half of a beta-carotene, which it is. We won't discuss the chemical and physical changes that happen to retinol as we consume it and as we use it in vision. Instead, I'll just point out that vitamin A is added to skim milk as vitamin A palmitate, so conjugated to a lipid. And that's because whatever vitamin A might have been present in whole milk is lost as we skim away the fat content of that skim milk. You'll also find vitamin A added to butter in Britain and your other European countries. Vitamin D3, cholecalciferol. This is a steroid-derived vitamin involved with calcium uptake and a variety of other things. But we're certainly familiar with vitamin D as being important for developing strong bones. And both, let's see, what can we say? So vitamin D3 is added to all milk products in the United States, and that was a development in the 1930s, I believe, to prevent rickets. So in the end of the 19th century, beginning of the 20th century, lots of vitamins were being discovered, and nutritional deficiencies were being found to be the culprits of lots of disease. And so we see the introduction of vitamins into our food supply. Vitamin A and vitamin D. Milk is not, I think, heavy in vitamin D3 on its own, but in order to find a food source that the entire population has access to and therefore help stem the tide of the disease, we add D3 to all milk products. And so vitamin D3 itself is not the active form of the vitamin. I show you on the bottom left here, cholecalciferol. That's the molecule that we consume in the milk. There's a series of enzymatic steps that leads to calcitrol. That's the active form. For the chemists in the audience, we'll just note that very simple changes have been made here. Well, maybe not so simple, but relatively small chemical changes have been made. A hydroxyl added here at the end of this aliphatic or fatty chain, and a hydroxyl added here. So again, modification of the milk product to suit social needs. Here's a snap from your text showing the composition of this oil and water emulsion we call milk. There are fat globules and proteins suspended in the solution. So what we see here are these hairy globules. That's the casein, supposed to represent the casein micelle. These little small squiggles are supposed to represent whey proteins, that is to say the proteins that are left behind when the casein precipitates out when we make cheese and other products. And then we see these enormous fat globules. What are these asterisks? Oh yeah, okay, so the asterisks are supposed to be reminding me to tell you that why is milk white? Well, it's because small particles, that is to say particles on the order of 200 to 400 angstroms, scatter light. That is to say they bounce light off them and change the direction of the light. And that means that what might otherwise be a clear solution becomes opaque solution. And because we're reflecting a broad wavelength range for visible light, what we see is white light. So both the casein micelles and to a greater extent the fat globules are able to scatter that light.